Hey everyone, thank you for coming today. My name is Mohammed Farhood. I'm the Director of Product Development at InVicro and working with and one of the members of the EMIT team. So InVicro are spinning out the company, and Robert Holt is going to speak on some of the implementation we put into VivoQuant related to tissue imaging and the Cryo Fluorescence Tomography. So Robert is senior scientist in the analysis group, but it's kind of hard because he does pretty much everything. You can see him in the lab, you can see him developing, so he's kind of a good jack of all trades. So he's here to present on what we've incorporated in CFT in VivoQuant. Thank you.
Thanks, Mo, for that introduction. Yeah, so today, an overview of the Cryo Fluorescence Tomography imaging suite in VivoQuant. So I know it's a mouthful, but we'll go over it on the slow side, I promise. So before we get into the software tools, I wanted to motivate what we're doing in the first place a little bit. Okay, so we have all kinds of ways of doing molecular imaging, and what we're looking at here is a comparison of the masses of tissue interrogated versus the resolution for a bunch of different imaging modalities. So down on the bottom left, we got all these microscopic approaches, confocal, micro photoacoustics, and what have you. And on the top right, we have in vivo fluorescence. So we have small things that we view a small amount of at high resolution and large things that we view a large amount of at low resolution. So this technology that we're talking about, where the software package fits in, is bridging this gap in between these two classes of imaging. So what is it? What is cryofluorescence tomography? CFT is a molecular tissue imaging modality where ex vivo fluorescence from individual 2D sections are captured and compiled into a 3D volume merged with anatomical white light reference. Very simple. So we'll continue to unpack this slowly. So the benefits are image sizes can range from cell clusters to entire bodies. So the resolution that we're talking about really is cell cluster resolution. We can image samples from rodent organs all the way up to like non-human primate heads, which is incredible. And we kind of, the technology that we're talking about isn't exclusive from other imaging modalities. It can be used in tandem with established imaging modalities through the use of VivoQuant, which I'll touch on again later. So this is what the images actually look like. This is, okay, this is, there we go. Just pretend like these all started automatically. So this is a view of fixed tissue. This is human tissue. On the left is a standard issue white light image as we continue to slice through the subject. In the middle is a fluorescence overlay, and then on the right is the actual fluorescence. So this was done with some of our collaborators at Stanford. So what we do is align all of these images. We do a little bit of optical correction to remove some of the subsurface fluorescence effects, and then we can render these things in 3D. So we have full 3D anatomical molecular information, but we also have this coupled to structural information in the context of what we see every day, white light images. Since everybody seems to need to have a chart, this is mine. This is the CFT workflow. So what we do is upstream, we prepare the subject. This can include administration of tracers. This can include induction of a tumor model that itself is fluorescent. This can also include longitudinal imaging in vivo. So it's possible to do PET, SPECT, diffuse optical, all kinds of medical imaging technologies upstream. Then once we do all of, so then on the right-hand side, the CFT workflow, the cryofluorescence tomography, we freeze the sample in a block, we section that block, that's what we were seeing before, sectioning and imaging of the block face. And then it's possible to collect some of the downstream samples for further analysis. So while we're slicing this, we can tape transfer something and then go do IHC or MALDI to it as a complementary imaging assay. And then we bring these things together using this VivoQuant, using these VivoQuant tools that we've been talking about all morning. So this is just an example of the sorts of things that we're talking about. And of course, as the larger we go, the lower resolution we get, but we get more valuable large information. On the left is a cryo slice image of a cynomolgus head. Then we're talking about a whole rodent body, and then resected rodent organs as we continue to go down. So we're talking about resolutions ranging from 50, 60 microns all the way down to about 20 to 25 microns. So I wanted to go over just a couple of images before we start taking a deeper dive into the software. These are a few examples. This is a comparison of a fly-through image for this is an AAV9 GFP that wound up accumulating in the liver on the right-hand side. And then on the left-hand side, we don't see very much in the control. Using some of our established software workflows, what we can do is multispectral imaging in order to remove some of the effects of autofluorescence. That was one of our big wins recently. So like I was talking about before, there's no reason why we can't include CFT into other established workflows. So what we're looking at here, the colorful stuff is fluorescence information, and it's co-registered to an in vivo X-ray CT. Because since we have this anatomical information, we have structural landmarks, which we can register directly to other structural imaging modalities using the VivoQuant software platform. Similarly, we can represent this white light data in a couple of different ways. One of them is a multi-slice view. So what we're looking at here is a glioblastoma model image with angiosense, blood pool agent. And right here we see 3D molecular fluorescence overlain on some 2D white light samples. This was done in VivoQuant. And then we can start to render these things in 3D as well. So we're looking at, in this case, is the same subject overlain on contrast-enhanced MR. So we get complementary imaging information. You can see the blood or the pool of the gadolinium there as well. Another thing we can start to do is time course type imaging. So the imaging technique is destructive. Once you slice through it, it's sliced up. But we can take a series of subjects and image them each individually. In this case, this was a time course of a labeled therapeutic in the brain. So again, since we have this anatomical information, we can use some of the VivoQuant tools to bring this anatomy into a reference space. We can do brain segmentations on these samples that we have, and then we can start to examine the time course of these images. And as I was discussing before, this is also complementary with, it's compatible with standard histology. So we can take a slice, transfer it, tape transfer it, or glass transfer, I guess, then stain it. So we can look at the same fluorophore that we were looking at in bulk, or we can also stain it with additional fluorophores to look at nuclei, to isolate the DAPI, or to do some kind of immunohistochemical staining on it. So I'm going a little fast, but I don't think anybody's going to get disappointed by getting to the break earlier, so I think we can come together on that one. All right, so this is an example of tumor metastatic spread that we did in conjunction with emit imaging. So what we have is a 4T1 mouse mammary tumor cell line that expressed both luciferase and DS-RED, immunodeficient mice, and this was an intracardial injection. So injected right into the heart, and then the metastases were allowed to spread. And we did bioluminescence imaging and CFT imaging. So this is what the CFT imaging fly-through looks like. So we can see a lot of metastases that have shown up everywhere. This is an image of the fluorescence. But we can also compare these. I mean, we can compare this directly to the bioluminescence imaging. I think this was actually done on the same day. So we did the bioluminescence imaging, and then we cut the subject up. So these, again, are complementary imaging assays. The bioluminescence can be done longitudinally in vivo, and we can compare it directly in VivoQuant to this high-resolution 3D fluorescence image. Even in the bioluminescence image, it's tough to see what's going on inside the subject, so sometimes nothing beats cutting it up and taking a look. This is a high-resolution example specifically from a tumor metastasis, in the femur. This is the sort of thing that is very, very difficult to see in many other imaging modalities, even in something like histology. But if you have a supercellular resolution imaging assay, you get a 3D view, and then you can comb through it later. And again, we have a lot of established workflows that perform image segmentation on whole rodent bodies. So we can bring that into the fold as well. What we're looking at here is, it's a little bit hard to see on this particular projector, but lungs, heart, and liver as well. And again, this was, I think, a flank xenograft that threw tumor mets into the body. This one, again, we can tape transfer a piece and then take a look at a microscope. This imaging methodology can also be used for multispectral techniques. So in this case, what we're looking at is 2 tracers in the same subject. One was injected intrathecally, one was injected intravenously. And in this case, what we have is a multispectral image. We have a comparison, a snapshot of the renal clearance comparing 2 different tracer injection routes. It's not shown, but we also have white light information that comes along with that kidney, which we can then use to segment. In this case, we use the white light information to segment the vasculature. So now we can see what's in the vasculature, what's in the medulla, what's in the cortex, what wound up in the ureter. We have this information at high resolution without having to rely on other structural imaging methods. So how does it work, right? That's the most important thing. So I just wanted to give a quick demo about what this software actually looks like in practice. Okay, so this is VivoQuant. We open a VivoScript. We have these image folders pre-prepared, and activate the CFT pipeline. We found some white light images. Yes, we would like to continue. They got brought into VivoQuant. We know from we know upstream what the size of the pixels are, so we can enter that. In this case, it was 75 microns with a 25 micron slice thickness. All right, so this is what the images look like. They look a little bit green because these are raw images off a white light camera, so the Bayer pattern augments in the green. They're not perfectly aligned, so what we do is we place some reference fiducials. So we know that there are these round black fiducials in the field of view, so we can place a few of them. There are 4 here. It's fast, but it's not that fast, right? So we painstakingly, by hand, place 4 fiducials. This is in real time, by the way. Clearly. We place 4 fiducials, which are then identified on all of the subsequent slices. So from slice to slice, we know that we're looking for dark round objects, and we can automatically find them for all of the slices that we're looking at here. So this green, so white light cameras tend to be extra sensitive in the green, and when you read directly from them, they appear artificially green. This is something that we're going to see corrected a little bit down the road. Okay, so quick QC step. Do these images have all of the fiducials identified where you think they are? Yes. So we keep it going. So this is talking about what kind of registration do you want. We support rigid and affine in case there has to be some sort of scaling and rotation correction that also has to be made. The application is fairly quick. There we go. So now we're checking. So this is after the alignment has been performed. How well does it align? Fairly well. Okay, so do we want to do color correction? The answer to that is yes, because we're dealing with raw images that came off of the camera. So what we do is look for a good background location. In this case, we know that the embedding material is white. It's uniformly white, and that's going to give us a good reference location. So we look, where is it the whitest the whole way through? It looks like that's about the right location. So we will paint. We'll paint a reference region, and this is the region that we're going to be calling our white light background. And there are two different methods of doing the white light correction. One we can scale to enforce that the background is white, and the other one is we can stretch to making the assumption that there is a dark location and a white location in the image, which generally is a pretty good assumption because the fiducial markers are black. So there we have it. So now we have aligned white light images of pretty good quality. In this case, the white light images were taken with the same camera as the fluorescence images, so they are natively co-registered to each other. That's how we get overlays for free. But we still need to take the alignment transforms from the fluorescence images, from the white light images, and apply them to the fluorescence images. So we load the fluorescence images, and we perform the alignment. And do we want to do background subtraction? Because there may be some stray light in the room. Yes. How do we choose a background region? We have a perfectly good one, but we can also draw another. Do a quick background subtraction. So this is applied to all of the images individually, in case the lighting conditions changed over the course of imaging for whatever reason. Looks like the background subtraction result is good. Do we want to do a median filter? No, the data is perfect as it is. Okay, do we want to do subsurface fluorescence removal? Yes. So this is the effect of imaging off the face of the block. We get some contributions due to subsurface fluorescence. So we see some of the fluorescence that's a few slices in. I love to talk about this sort of thing, but it's sort of outside of the scope of this particular manifestation of the software. So what we do is we choose parameters based on the tissue type and based on expert experience for how do we best correct for this fluorescence. So it amounts to a sharpening of the fluorescence, a removal of some of the blurring effect that comes from having block face imaging. The correction is performed in the filtering operator in VQ. Okay, do we want to do deconvolution? In this case, no. So if the user has a measured point spread function that they have, then they can introduce that at this stage, and they won't have to do subsurface fluorescence correction. If they know the point spread function of their system, they can just remove it. They can add it themselves and remove it from the data set. So do we want to do visualization? Of course, it's VivoQuant. So we're going to zoom in. We're going to zoom in on this bottom animal and just take a closer look at this one in particular. So we crop. It resamples to isotropic voxels, and now we can do a quick review again, and here you can see the fluorescence overlain on these corrected white light images, and then a 3D view as well, which I believe Gabe touched on earlier, with white light overlain with fluorescence. So in this case, the bright embedding material has been removed, but the fiducials remain. But it's looking pretty good. It's not bad for what, 8 minutes of work. Don't tell my boss I said that. So now we can Now we can adjust the knobs a little bit, play with these images, try to render it in a little bit more of a pleasing fashion. So from here, we can adjust the knobs. This is after I played around with it a little bit to remove the fiducial markers, to turn the knobs and adjust the opacity until it looks really nice. And we can adjust the opacity on the white light images. We can change the fluorescence images, all to enhance different aspects of what we want to be seeing in this particular case. So that is a little bit fast, a little bit early, but that's the long and short of how the image process works. And with that, I'd be delighted to field a few questions if you have them. No, so the slice thickness is variable, and it can be whatever the user desires. We've gone anywhere from, I think, about 15 to about 50 micron slice thickness.
And have you worked with a standard pathology slice at 5 micron?
So the question was, do we work with 10 micron images? And the answer to that is the answer that I just gave. So do we work with standard 5 micron pathology slice thicknesses? Yes, we have also worked with that. So in that case, it kind of depends on what the desired workflow is. We can slice through an entire sample at 5 micron, or what we can do is, if we're slicing through at, say, 25 micron, and we want a 5 micron slice, we can cut a 5 micron slice and then a 20 micron slice, and then just pick up where we left off. So the instrument that we have has a big pause button. So these images, as they're coming off the instrument, you can see them. So a pathologist might want to say, okay, let's stop. That's looking fairly interesting. I'd like to see that at a little bit higher of a resolution. Tape transfer slice, then cut a 20 micron slice, and then sort of press the resume button to keep going.
Yeah, I have a slightly design-related question. So let's see where they have a fluorescence ligand where you need iris image, where you need X-ray or tissue level image. We know what amount that we inject. And if you do this biosectioning, if we want to say one gray region, every gray region should be above the background. We know sinus or cos or something. We want to see the biggest signal. We roughly know it's there, but what amount should I adjust my those ligand to get a good this 3D section?
So I believe the question is, how much do we have to adjust the dose to get a good image? I think there's a bit of an art with that one, but what we have found is, if you can see it on an IVIS, you can definitely see it in this imaging modality. Our sensitivity, I believe, is down in the, I think it's safe to say, dozens of nanomolar range, but it's probably lower than that. So if you know the concentration, then if you know the expected concentration, then you can adjust the dose accordingly.
So we have one question from online. It says, are you able to perform corrections such as spectral unmixing, specifically looking at GFP imaging?
So yes. So this is the question is about, can we perform spectral unmixing? And the answer to that is, based on our instrumentation, that technology is still fairly young, but we can do it. So the filtration is done, the way the imaging is done is by notch filtering. So if we have a reference channel and an active channel, say we have a GFP and then either a red channel or a blue channel image that can be a surrogate for the autofluorescence, then yes, we can do spectral unmixing to remove that. But do we have hyperspectral techniques to do multifluorophore spectral decomposition? We're not quite there yet, but hopefully someday we will be. Okay. If nobody has any other questions, then thank you.